Good evening. We'll begin the class now. So we'll look at sample problems for week 10's experimental biotechnology course. So to start with, the first question is dash is a pyrimidine base commonly found in DNA. Four options are given here: adenine, xanthine, uracil, and thymine. So whoever knows the answer for this can take a couple of minutes to think and first differentiate what pyrimidine and pyridine is. There are two different types of bases that are found in DNA. So once you differentiate the structural differences, then we'll look at the pyrimidine base. Yeah. So Sujita, you can unmute and tell what are the different bases that are found in DNA and what are the names of the bases that you are aware of, the classifications and the names. You can unmute. Okay. What are the differences between purine and pyrimidine? No, no. So what is a purine? Okay. So in DNA base pairing, do you said adenine and thymine. So do you also know how do they interact? Do they form bonds? Hydrogen bonds? Yes. So which of these two will form double bond? Right. And okay, that is correct. Now, does a purine and a purine form a double bond or a triple bond? Or is it always the case that purine and pyrimidines form bonds? Can you explain what you're trying to tell? Okay, so we have two different kinds of bases in DNA. I mean, in our body. So one is purine. What you see here as a six-membered ring and additional three members, six plus three. This is a purine. So adenine and guanine belong to purine, whereas cytosine and thymine are uh, pyrimidines. And in addition to this, you also have uracil, which has one more modification to these the cytosine and thymine. So, and it's always the case that one purine will hydrogen bond with another pyrimidine, and that is how it is observed in DNA. In RNA, you have different cases. So we'll not get into that as of now. But since you're talking about DNA, we need to. This is what we need to know. And among these, pyrimidines are the ones with the lower number of rings, which is cytosine and thymine. So among these different options, thymine is the right answer. So thymine is the pyrimidine base commonly found in DNA. That is the appropriate option. And other than this, do you know what xanthine is? No, no. So xanthan is also similar to your purine. It also has this double ring, and this is an intermediate that is present. We don't have xanthine as one of our bases, but it is present in our body. It doesn't form in the DNA. It has some chemical modifications compared to this. Right, right. So that's also incorrect. Fine. The next question is dash is not true regarding the structure of DNA molecule. Four options are given here. One strand of DNA dictates the sequence on the other strand. Two strands of DNA run in the same direction. Base stacking provides stability to the DNA structure. Ratio of purines and pyrimidines in DNA is 1:1. You can think about this for a couple of minutes and then we'll discuss. Okay, do any of you know what base stacking is? No problem. So what you see here is a base pair, that is a pyrimidine and a purine and a pyrimidine will hydrogen bond. Similarly, you will have one below and one above them. If you see the nature of these rings, these have delocalized electrons within them, which allows them to interact in a pi-pi manner and gives it more stability. This is the base stacking. So this interaction gives additional energy that ensures the DNA structure is more stable. And finally, the last option is one strand of DNA dictates the sequence on the other strand. This is again because you have this base pairing complementarity, and adenine can interact will base pair with thymine only. So if you have on one sequence adenine in a particular position, on the other sequence you know you'll have thymine. So if you know this sequence from one uh, orientation, you'll be able to tell the sequence of the other orientation based on base pair complementarity. That's what this option refers to. Fine. The third question is DNA synthesis occurs in dash phase of the cell cycle. M phase, G1 phase, S phase, G2 phase. So you can think about this for a couple of minutes and then start by describing whatever you know about the cell cycle. Whatever you know about uh, cell cycle, maybe we'll take it from there. Its phase synthesis, that's correct, but we'll also discuss in general about the cell cycle. Do you know about other phases in the cell cycle? Okay, that is at the molecular level. Uh, yes, you go ahead. Okay. Hmm. Hmm. Okay, these are the molecular level, correct? Yeah, you have the DNA polymerase, you have the single strand binding protein that your helicase will first unwind. Your single strand binding protein will ensure it doesn't come back. Then you have primers, Okazaki fragments that will act. for you to make this replication. But let's not go into the molecular detail now because the question is about cell cycle. So, do you also know something about the cell cycle? Yes, Nityashri. You can also try answering if you know something. Sujita or Nityashri, one of you, either of you can answer. Oh, right, right. And do you know what happens in each of those phases by any chance? In all of these, like generally. So, what happens in M, G1, S, and G2? You had defined what happens in mitosis. You have the interphase, metaphase, sorry, interphase, prophase, metaphase, anaphase, and telophase. You're right. So, what we also, what I generally wanted was this. So, in the G phase, you have this cellular content excluding the chromosome that is duplicated, which means everything other than your DNA. In the S phase, your chromosome is duplicated. So what you see as these single strands get duplicated. Then in the G2, the cell will check the chromosome for error. Then you have this prophase, metaphase, anaphase, and telophase where these distribute or the chromosomes which are duplicated will be distributed to two cells. You also have a G naught phase where this cell cycle can go into arrest. Fine. Good. The next question, it's not very biology related, but it's also we use an application of this. So I would rather explain this. In thermal cycler, Peltier phenomenon occurs at dash and N-type semiconductor, P-type, either N or P at the junction of P and N. So um, instead of looking at this question as a purely electronics question, does anyone here know what a thermal cycler is and where it can be used? PCR. Okay. Why do you need a thermal cycler in a PCR? No, no. Yeah, So if it is only amplification, you need only one temperature, right? Why do you need a thermal cycler if you need an amplification? Okay. Do you know the stages and if possible the temperature as well by any chance? You can try. We'll discuss and we'll come to a final answer. You can tell me what you know, we'll take it from there. Right. So what will be the temperature approximately there? Yes. 95 is sufficient. Yeah. Right. Right. Mm-hmm. Mm. What is annealing? Right. So this will have a different temperature other than 95 degrees. Right, right. And after annealing, what other step do we have? Right. And so here, what is the temperature that we use? No, so the timing here would vary because timing depends on the length of your amplicon and the rate at which your polymerase enzyme can amplify. But again, the temperature also depends on the maximal activity of your um, polymerase. People generally use stack polymerase or fusion, and for both of these, 72 degrees Celsius works. Right. So because we have this variation in temperature and this goes on for multiple cycles, we use something called as thermal cycler. And here, the way in which this temperature can be changed is based on this Peltier effect. So here, by applying voltage between two electrodes of the semiconductor, the temperature difference can be achieved. So this way, we'll be able to transfer heat from one medium to another. For this, we need this junction of p-type and n-type semiconductor. So although you need not specifically know about the details of the working of the thermal cycler, we need to know at least where we use it. So good. You guys are aware of it. Good. The next question is dash property of Taq polymerase enzyme makes it useful for use in PCR technique. Option A, it does not require primer for DNA synthesis. Option B, it can use RNA as a template. Option C, it is stable at 95 degrees Celsius. Option D, it has proofreading activity. Fine. You can think about this question for a couple of minutes and start by telling me what you know about TAC in general. If you know the full form, you can even start from there. That is still useful. Yes, so does anyone want to uh, tell what you know about Taq polymerase? Hmm. Right, right. And why do you think thermophile polymerase can withstand 95 degrees? Right, yeah, that's the logic. So thermophiles grow in these very hot temperature conditions, like for example, hot springs and so on. So the enzymes in thermophiles are generally known to be stable, uh, and easier to crystallize as well. I mean, stability of an enzyme can be correlated with crystallization ability as well. So if you observe, many homologs of thermophiles are usually crystallized when you don't have a model system characterized structurally. This is majorly due to its rigid core. Again, at the end of the day, these are just proteins. So the more rigid their core is, the easier it is amenable for structural studies. So that is one characteristic feature of thermophile enzymes and proteins. And yes, like you mentioned, because of its stability at 95 degrees Celsius, we do use this as an enzyme for in our PCR reactions. And yes, it cannot. It although this is uh, this is stable, it still does require primer. So option A is wrong, and it cannot use an RNA as a template. And proofreading activity, all yeah. So this proofreading activity is usually conferred by enzymes with multiple domains or multiple chains. So what is observed is once the chain is elongated, it the other chain or the other subunit in the enzyme will go back and it will check whether there is enough fidelity. That is, whether a purine or pyrimidine, if it has been incorporated rightly. And so, yeah, uh, although this is right, at least in the commercially available Taq polymerases, the feature that makes it helpful for us is its stability at 95 degrees Celsius. The next question is DNA synthesis occurs in option A 3' to 5' direction, option B 5' to 3' direction, option C both 5' to 3' and 3' to 5', option D either 5' to 3' or 3' to 5'. Now, before you answer this question, you can think and you can tell me what will dictate the directionality for DNA synthesis. We'll take it from there. Replication as in, do you think is there any molecular component that will dictate the directionality? You have a DNA, you want it to replicate. Now, is there any molecular component that will decide whether it has to happen in 5' to 3' or 3' to 5' or either of it? Right. So the nature of your DNA polymerase is what will dictate the directionality. Right. That's correct. So now let's come to um, the, what is the bond that is being formed when DNA synthesis happens? Yes. So, and this phosphodiester bond happens between what and what? Yes, correct. So now for this to happen, you need to have your 3' OH positioned well, whereas your 5' phosphate can be at least slightly freely floating. This is what dictates your directionality. Your enzyme can do it from uh, 5 prime to 3 prime because your 3 prime OH and because your 3 prime OH should be more or less rigid, whereas your 5 prime phosphate can be slightly flexible. If it's the other way around, it cannot happen. So this is what directs the directionality. Right. Good. And just one more thing that I would like to add here is in DNA replication, what not in PCR terms, but in how it happens within the cell, you have something called as a leading strand and a lagging strand. Now, in one of these, you have a primer and that gets elongated. In the other case, you have discontinuous replication where you have something called as Okazaki fragments. So these get synthesized and you have these smaller fragments which later on the DNA replication machinery would take care of. Fine. Yes. Now this is a question that we discussed earlier, but still we'll just have a small recap. Taq polymerase is obtained from option A, Thermus aquaticus, option B, Pyrococcus furiosus, option C, Escherichia coli, option D, Saccharomyces cerevisiae. And from the name itself, you can get it. So usually these enzymes are named after the genus and the species name. For example, here TAC is can be broken down to T from Thermus and AQ from Aquaticus. So Thermus Aquaticus is nothing but a hot spring, water, natural hot water springs. This is where this organism survives. Okay. The next question is dash is not required for amplification by PCR. Magnesium ions, thermostable DNA polymerase, helicases, primers. You can take a couple of minutes to think about this and then justify first the incorrect options. Then we'll go to the correct option. Okay. Hmm. TAC is one thermostable DNA polymerase, one of the thermostable DNA polymerase. Yes. Why why do you think helicases is not required? Um, so what do we do if we are not going to unbind DNA? Correct. So instead of using another enzyme called as helicase, what we can do is we can play around with the temperature, and at 95 degrees Celsius, we expect all these hydrogen bonds to be completely off. So the strands get separated and our primers can anneal. That is the logic. Right. That's why helicases is not required for amplification by PCR. You're right. Good. The next question is, how many different primers will be required to amplify a 400 base pair segment of DNA by PCR? And four options are given here: 4, 2, 1, and 8. So, in this question, uh, one option is definitely incorrect, and there are three options which can be correct depending on the context. So, does anyone know what that one option that is definitely incorrect? Uh, why do you think 8 is incorrect? Yes, yes. We, yeah, yes, that is right. So 2 is definitely correct. And what I was trying to get across is one option that is definitely incorrect. That is option C, which is 1. With one primer, you cannot do amplification. If you do amplification, you will get a single strand. You will not get a double-stranded DNA. Whereas options 4 and 8, although it might appear like these are wrong, you can still have four primers at different regions and still be able to get amplification, or eight primers. The only thing is, where do they sit on the DNA? So there we have methods called as overlap extension PCR and other PCR related methods where we use more than one primer, more than one pair of primers. So, although for this particular question, 2 will be right, even 4 and 8 are applicable if we are talking about other cases of PCR. That is what I wanted to get across here. So, here we have a primer sitting on either end of the uh, uh, either strands, and then it amplifies. So, what one needs to understand here is each molecule of primer will get consumed in the process in one cycle. So what was once a primer is now a strand. So it can no longer be able to bind to another primer, another actual strand. Rather, this strand will act as a new base for your primer to bind. So with this, with 20 to 30 cycles, we have millions of copies. The next question is, optimum temperature for Taq polymerase is dash 95, 37, 72, 55. So yes, this is the extension temperature. And since we're talking about polymerase, we need to think about the activity, which is the ability of it to polymerize a chain. So at 72, it has maximum activity. The next question is, after 20 cycles of PCR using PFU DNA polymerase, dash number of molecules may have mismatched base incorporated in them. So here again, two options are definitely incorrect. So first you can start with telling me what these two definitely incorrect options are. Then we can go to the right option. Yes. So in a PCR you cannot have one in every three base mismatch or one in every five base mismatch. So definitely options B and C, 33% and 20% are incorrect. This 3.5% or 2.5%, it depends on the polymerase that you use. So you can refer to specific manuals. But from what I am aware of, this PFU is also very high fidelity. This is what people sell as fusion. This is from this organism called as Pyrococcus furiosus, which we just came across a couple of slides ago. Pyrococcus furiosus. The next question is, after amplification by PCR An extra spurious band was observed along with the desired band. This problem can be solved by dash. Increasing magnesium concentration, increasing enzyme concentration, increasing the number of cycles, increasing the annealing temperature. Okay, now um, you can take a couple of minutes to think about this. So, first, um, one can tell me what they understand by an extra spurious band along with the desired band. Once I know that you're able to understand the question well, then we'll discuss why this could happen. You can take a couple of minutes. Yes, so you have one primer, you have one template, but because you have multiple possible, um, ideally your primers are okay. We have been going on discussing about primers, but does anyone know how a primer is designed? Right. So, do you know how primers are usually designed for PCR work? Hmm. Yeah, like any parameters that you would specifically look for? Yes. Yeah. So, do you know of any of these characteristics? If you do not know, it's still okay. So what I'm trying to show here is how we generally design primers. So what you see here is this is a plasmid map. This is of a protein called eGFP. This is just enhanced GFP. GFP is green fluorescent protein. So now let's say this is what I want to amplify. So what I would do is I would select a few bases in such a way that the melting temperature, what you see here as Tm given in purple, is 57 to 59. Ideally, what I do is I choose from 55 to 60 degrees Celsius. And then this is what we choose as our primer of interest. This is one primer. Okay. So we ensure that this is one primer. And let's say I want to amplify this EGFP gene, which is given as green. I go to the other length till the stop codon, then go to this and then choose the number of bases in such a way that the other TM is also 55 to 67. So if you had noticed here for 22 bases, although it's shown as base pairs, for 22 bases it is 56 to 57 degrees Celsius. Whereas here in the forward primer, for 16 to 17 bases itself, we have 57 to 59. So ideally, what you should optimize for is the length of, sorry, is the temperature. And if as long as both of your primers are in the similar temperature range, your experiment would work. Was that clear? Okay. So now that we had discussed about this primer, yes, yeah, Sai Sampath, you were telling about annealing temperature. So this the temperature in which we design that's the annealing temperature. So although we will be able to design this with higher accuracy, in some cases we'll get these other bands. That is because you have some partial overlap, maybe at the 5' region or at the 3' region, majorly at the 3' region, because even if you have it at the 5' region, and uh, only a few bases can are common between two different regions at the 5' region, since extension has to happen from 3' region, your enzyme will not be able to sit on the primer and start with DNA replication. So uh, that's the logic. And now, because this is happening only at some bases, not all bases, by increasing the annealing temperature, what we can do is we can ensure that this non-specific binding can be reduced. Yeah, that's what you wanted to convey, right? Sai Sampath? Right. Good. Okay. The next question is, contamination of Staphylococcus aureus in meat products can be detected by PCR using primers for the dash gene. And four options are given here: NUC NUC gene, black Z, FA, ORF1. You can take a couple of minutes to think about this, and to start with, you can tell me what you know about the incorrect options. You might be familiar with the incorrect options and the expansions for them. We'll start from that. Okay, you can first tell us what you know about the incorrect options, and then we'll go to the NUC gene. Okay, Laxi. No, it can't be in just uh, Enterobacteriaceae. Yes, I Sampath, you were talking about Lacoperon. Have you heard of Laxi? Yeah. So, what do you know about it? You can tell us what you know about it. We'll take it from there. Okay. So, like you mentioned, it's a part of Lacoperon. You have in an operon, you have multiple genes. So here you have Lac ZYAIPO, of which P is your promoter, O is your operator, I is your repressor. So your ZYA codes for different genes that allow for entry of lactose within cell on its breakdown. So lactase is present in multiple organisms, not in Staph aureus alone. And like you mentioned, uh, yeah, like Sujita mentioned, Ceph A is in Salmonella. And ORF1, any idea? ORF is actually, I'm not sure if it's present in only one. ORF is nothing but open reading frame. So it is very, very non-specific and a very vague or a very general name that need not be associated with one particular species, like Laxi. So whereas NUC is this encodes for a nuclease that is specific for Staph aureus alone. Fine. Right. The next question is dash is used to classify plant species using species or group specific primer in PCR. Option A genomic DNA, option B mitochondrial DNA, option C mRNA, option D chloroplast DNA. Now, before we go on to the right answer for this, if you guys are aware of the wrong answers, you can tell me or justify why a particular option is incorrect. Based on that, we'll uh, decide why a particular option could be correct here. Right. Okay. Hmm. Mitochondrial DNA is present in every organism that can have a mitochondria. So, what is the kind of organism that can have mitochondria? What kind of organisms will have mitochondria? Right. Good. Hmm. So again, you'll not be able to distinguish within eukaryotes uh, based on this, right? Hmm. 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 Right. Yeah. Uh, Basically, that explains. Yeah, chloroplast DNA, again, plastid DNA is only present in plants. So with that, we'll be able to classify plant species. If we were to look at, let's say, prokaryotes, what do you think would be the right uh, marker to identify a particular prokaryotic species? Any idea? Okay, no issues. Others, Nityashri and Sujita, any idea as to what particular species will help us differentiate between different prokaryotes? Sorry, what viral? No. I'm asking what particular DNA stretch can be uniquely present in each prokaryote. Okay, no issues. So, have you heard of the ribosome? Any of you? Yes. So, do you know what this S corresponds to? Right. Yes. So this just tells us about macromolecular assembly. So the ribosome is a very large complex. It contains multiple proteins and few RNA. These are called ribosomal RNA. You would have studied the different classifications of RNA: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. So these ribosomal RNA are specific. Some of these are specific to species. So even in E. coli, when we do experiments, we use some uh, experiments where we quantify the gene expression. We use something called as 16S rRNA, which is a universal marker that is used for quantification. So this is also specific to species. So, like we mentioned about chloroplast DNA for plants, for prokaryotes, we can still use this 16S rRNA as a marker. Good. And the last question is typical concentration of DNTPs in a PCR reaction is, and here if you see 200 millimolar is a lot, 200 nanomolar is very little, 200 molar is extreme. Your cell can't withstand such a large concentration of salts, and 200 micromolar is what people use for normal PCRs. Now, a quick addition to this. Now, let's say I want to introduce. Okay, when I say 200 micromolar of DNTPs, what do I mean? Does anyone want to try answering that? How much of each nucleotide is present when I said 200 micromolar DNTPs? So I'll repeat myself. So what I asked is when I say that my Eppendorf tube or my vial has 200 micromolar of DNTPs, how much of each nucleotide do I have? How much of DATP, DGTP, DCTP, and so on? How much of each of these do I have? That was the question. Does anyone know, have a broad idea? No, no, I'm asking about this nucleotide. Uh, so in total I have 200 micromolar. Now we have four of these nucleotides. How much of each of these nucleotides do I have? That is the question. Yes, yes, that is right. Right, good. Now, when will I use more of one particular DNTP? I mean, one particular adenine or cytosine or thymine or guanosine, and less of another one. When will I use different concentrations of this? Any idea? Or what will happen if I use different concentrations of these and not equal concentration? Yeah, yeah. So we are discussing about PCR only, but let's say I have different concentrations and unequal concentrations. What will happen? Yeah. So how will that imbalance affect your PCR? Yes. So Yes. So will your reaction stop or what will happen? Okay. So what people, when do people generally use this is when they want to introduce mutations. So they reduce one particular nucleotide. As a result of this, these nucleotides get randomly incorporated, giving us random mutagenesis. This is done on purpose to be able to identify if, based on random mutagenesis, do we get any good variations. So people do use this. That is when different concentrations of nucleotides are used. Yes, yes, true. In Sanger sequencing also we do that. There it gets stopped. But one variation there is that is a double modified nucleotide where extension is stopped. If you remember rightly in Sanger sequencing, I think it's not deoxy, it is dideoxy or something. So your extension will stop. Here we don't have that modification. So what will happen is your nucleotide will get incorporated. So you get changes at the protein level. At the DNA level, you will still get an amplicon of the same size. But at the protein level, you will see differences. Your protein might have a different mutation or a different frame shift altogether. Most probably, it shouldn't have frame shifts, but that might also happen. And this will give rise to new variants of protein that you can work with. Fine. Was that clear? No, no. So here in this, okay, in the question that I asked, when we use different concentrations, it doesn't stop. You will get your full amplicon, but you will get some mutations because that is the purpose why you use different concentrations. There you will still get your full amplicon, but your amplicons will be of different uh, sequences. Your amplicons will not be 100% identical. You will have mutations in some places, which you will know when you sequence the clones. So no, you will get, so amplification just means your base is being incorporated. So there is no, so the amplification is happening as per it should happen. But because you have less of one particular base, you will have mutations in some positions. And this will be random. This method is random mutagenesis. One of the random mutagenesis methods. Did it make sense? Oh, okay, good. Yeah. So with this, we come to an end for today's set of questions. If you have any questions, I'll be here to answer. Yes. Yeah. So let me open that 1 minute. So you have different names depending on what people use. I can show you one example here. Give me a minute. Okay. So you see this, right? Now let's say this is one of my primers from here to here. Now let's say I add this as one of my primers. Okay. Now let's say from this till this region it is happening. Now if I add one more primer here, what I'm trying to tell is I can add one more primer here. Again in the top strand, this is again primer 2. This will be my primer 3. This will be my primer 4. So what I'm trying to tell is now, let's say you have all these four. Wait, 1 minute. Yes, you have all your four primers here. Leave this one. Let me delete this. You have all these four primers here. Now, when you amplify this to this, let's say you use all these four primers. This primer can combine with this also. This primer can combine with this also. One forward primer can sit on one stand and amplify the other. So, depending on your extension time, you will still at the end, because you have a 5-minute extension, your length of the amplicon will still be from this to this, although you have intermediate primers. People generally use these intermediate primers in a different way. Like, for example, instead of having this primer from within, they use it in a um, different setting. For example, a primer like this will be designed here, and you will create something called as an overlapping region. And for example, here, let's say we go to this primer. What we do is let's say we add a overhang here. Now, let's say we've added an overhang here. Similarly, let's say we do from this somewhere else region. So in those cases, multiple primers can be used. This multiplex PCR, I think, is also one such method where you can use multiple primers to increase the length of your amplicon. Right. Yeah. So now, what was your question? I mean, the minimal set of primers that you need is two. You can have as many primers as you want for amplifying. Yeah. Okay. Good. Sorry, calculate as in? So you will never be able to calculate it before you get your PCR done. The only thing that you can do is once you finish your PCR, you can run it on gel, and based on the intensity of your product, you will get an idea. One more method that people use is absorption at 260 nanometers wavelength. So based on that, you will get a particular concentration, usually in nanogram per microliter. So based on that, you will calculate your the amount of DNA that you have in the eluted volume of nucleus-free water or your TAE buffer. Thank you. Thank you.